Phenotyping the locomotion of C. elegans is an important tool in the discovery of genes and neurons involved in C. elegans behavior. C. elegans undergo stereotyped locomotory behaviors such as forward and backward motions and various types of turns by changing their shape. To quantify these movements over long distances and at high spatial and temporal resolution, we have built a worm tracker to capture the motion of C. elegans as it crawls on the surface of an agar plate. We have also developed software to isolate and analyze these movements quantitatively using custom programs written in LabVIEW and MATLAB.
A challenge of our video tracking system is that they generate an enormous amount of data that's highly dimensional. Our image processing and data analysis program deal with this challenge by reducing the worm shapes into a set of independent components, which have comprehensively reconstructed the worm behavior as a function of only 3 to 4 dimensions.
An agar plate is illuminated by a fiber light source and imaged with a camera. This system is mounted to an XY translation stage. The stage is moved by standard stepper motors, which are connected to a stepper motor controller. The controller and camera are connected to the computer and controlled by custom programs written in LabVIEW. The camera images the surface of an agar plate and identifies dark objects on a light background. A calibration mark for the automatic calibration process is made by poking the agar surface with a worm pick. The image quality is adjusted so that the computer program can quantify objects in real time.